Hello, everyone, and welcome to our MaxCyte sponsored webinar titled Scalable Manufacturing and Nanovesicle Delivery of CRISPR Cas9 Ribonucleoproteins using a cGMP compliant cell engineering platform. My name is Leslie Eschinger, and I am a Director of Marketing for MaxCyte. I will be your moderator for today's engaging session. I would like to remind everyone that this event is interactive, so we encourage you to participate by submitting questions at any time during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If at any time you have trouble seeing or hearing the presentation, please click on the tab found at the top right of the presentation window. Our speaker for today's session will be Peter G., a Field Application Scientist at MaxCyte. For Peter's complete biography, please visit the Biography tab at the top of your screen. Without further delay, let me turn this session over to Mr. Peter G.
Okay, so thank you for the introduction, Leslie, and thank you to everyone who's tuning in for today's webinar. Before I joined MaxCyte, I was a postdoctoral research fellow at the Center for iPSC Research and Application at Kyoto University in Akitsu Hotta's laboratory, and there we were working on developing new technology, delivery technologies for the CRISPR Cas9 ribonucleoprotein complex. So today I'm really happy to share the results of our research. This was 5 years in the making. It was recently published in Nature Communications, showing the development of a new CRISPR Cas9 delivery platform using a GMP compliant cell engineering platform. So for today's agenda, I'll go into the background about viral versus extracellular vesicle delivery, and then talk about the development of our RNP delivery system that we term Nanomedic. I'll then talk about a few applications using Nanomedic in various cell types, some difficult to transfect typically, and also some in vivo studies where we show delivery of Nanomedic in vivo. Then I would like to go into the development of Nanomedic by electroporation. We found initially that we could generate Nanomedic at the small scale using lipid transfection, but we found that we were having struggles with scalable production of Nanomedic. So we then moved on to Nanomedic production by electroporation, and then scalable production by flow electroporation. So going into the background of CRISPR Cas9 and its delivery, I think most of you may know, but CRISPR Cas9 is a powerful genome editing tool to introduce double-stranded breaks into the host DNA genome, and it consists of a nuclease and a guide RNA molecule that can identify a 20 base pair complementary sequence in the genomic DNA. So the only difficulty with applying CRISPR Cas9 for therapy in the clinic or in difficult to transfect cells is there are very limited options for delivery of this system. So adeno-associated viruses, you can see on the right figure, is the most common vector for delivering CRISPR Cas9 in vivo. And it expresses Cas9 in the guide RNA through an episomal DNA vector. The expression can be very long-term, actually years being expressed from this vector, but there are a few disadvantages. So those disadvantages are an increased risk of an immune response to, for example, the Cas9 protein itself being expressed in the corrected cell. So even if you get correction of your cells, if they express Cas9, they could be eliminated by your immune system. And second, the fact that you're using CRISPR Cas9 to introduce double-stranded breaks, sometimes the viral vector backbone will be non-specifically integrated into the host genome. So there is the risk of that. And finally, there's a risk of off-target cleavage by the CRISPR Cas9 nuclease. So if you leave it in the cell for too long, it can cut similar site recognition sites that may be off by up to 4 base pairs. So ideally, you want to deliver CRISPR Cas9 transiently for it to do its job and then get it out of the cell. So we we sought to develop a transient CRISPR Cas9 delivery system that we hope for, that we could apply for in vivo gene editing. And so to do this, we kind of looked at extracellular vesicles as possible delivery vehicles. So what are extracellular vesicles that I'll refer to as EV from now? So they're cell-derived membrane structures that range in diameter. It's pretty diverse, but the ones that I'm referring to are 50 to 100 nanometers in diameter, and they're secreted from cells. So previously, these secreted EVs were thought to kind of be the garbage trucks of cells. They carry excess cargo, which is unnecessary. That's what was believed before. But actually, the cargo that they carry are bioactive molecules such as lipids, proteins, and RNA that have biological function. It's involved in immune responses and intercellular communication with other cells. And sometimes viruses can use them for viral pathogenesis. So if you kind of look at the figure on the right here, this is a cell infected by HIV, but some viruses, when they overexpress their proteins in cells, they can utilize the EV machinery to package their own proteins and components into these budding EVs. So in a sense, you can think that if you overexpress a protein of interest in a cell, you can utilize the EV machinery to deliver your protein of interest. So you can kind of use these EVs as suitcases and package what you want into them for delivery into other cells. So in this sense, you can harness EVs for protein delivery. And EVs can essentially be shells of self-assembling viral structural proteins, and you can remove the genomic DNA and genomic RNA. So just as an example, we wanted to look at lentiviruses as kind of a base for this system. And lentiviruses have enzymes such as reverse transcriptase and protease and integrase, which are involved in various steps of the virus life cycle to permanently integrate DNA in the target cell. However, if you kind of take away these components and just use the structural aspects of viral particles, such as an envelope protein for cell targeting, GAG, which is a structural protein which helps kind of the particle formation, you can just use these two components, and we wanted to package the Cas9 protein and the guide RNA into these engineered EVs. So in doing so, what we aimed to do was, well, we wanted to deliver the CRISPR Cas9 complex transiently. So if you were to deliver it by lentiviral transduction, which a lot of laboratories do, the lentivirus would bind to a target cell, be endocytosed, and release its contents. So the viral RNA genome would be reverse transcribed into DNA and integrated into the host genome. And then you would express your Cas9 protein and guide RNA, and then you would get genomic DNA cleavage. However, again, long-term expression of Cas9 is not really desirable, and we don't want to leave it in the cell for a long time. So if you look to the right schematic and you look at transient delivery by EVs, you can still utilize the receptor binding and endocytosis and release of the contents. But when the contents are released, they're already in its RNP format, so it can immediately cleave the genomic DNA target of interest. And after that, the protein's degraded. So hopefully you can get high activity of on-target cleavage, but the RNP complex would be degraded, so you reduce the risk of off-target cleavage by Cas9. So to develop this system, I'll save you the trouble of looking at negative data, and I'll go into the system that actually worked for us in the end. We decided to utilize a chemical ligand-induced dimerization system to package Cas9 into our particles. So if you imagine this is a producer cell, so we're producing our EVs by utilizing an HIV GAG protein, which is fused to a dimerization domain. So GAG localizes to the plasma membrane, and I'm only showing one GAG molecule here, but up to 5,000 GAG molecules can be incorporated into one particle. So what we did was we introduced a cell-permeable chemical ligand into the cell, which would bind to the dimerization domain of GAG. And at the same time, Cas9 is expressed and also fused with the dimerization domain. And this dimerizer will recruit Cas9 into the budding particle for release. So in doing this, we could specifically package the Cas9 protein. So when we produced these EVs containing Cas9 protein, we delivered them onto a GFP reporter cell line that already expressed guide RNA. And if Cas9 is delivered, what happens is that this target sequence is cleaved and GFP expression is restored. So if you get higher GFP expression, that means that Cas9 was delivered into the cell and you obtain genome editing. So you can see here on the figure on the right, bottom right, is that with the chemical ligand introduced, you have a much higher degree of GFP positive cells as opposed to no chemical ligand during the formation of these EVs. So this indicates that Cas9 can be stochastically introduced into these particles by overexpression, but specific incorporation using the chemical ligand really enhanced the packaging of this Cas9 protein. So the next part of this study for the development of our EVs was packaging of the guide RNA. So we really struggled with this, and we tried a lot of different published approaches that didn't work for us. So what we ended up using at the end was a system where we would have the guide RNA flanked by self-cleaving ribozymes indicated on the top with the DNA vector. And this is preceded by a Cy packaging signal. So what happens is that the DNA is in the producer cell is transcribed into RNA, and the Cy packaging signal is it binds to the GAG protein. So in the budding vesicle. So similarly, like you have Cas9 recruitment through dimerization, but GAG is also recruiting this RNA molecule into the particle. And once the RNA molecule is recruited, the ribozymes self-cleave, and then the guide RNA is liberated in this all-in-one EV. So if you look at the bar graph on the right, what we did was we did incorporate guide RNA into these EVs, either using the packaging signal or not using the packaging signal. And you can see, again, some of the guide RNA can be packaged non-specifically into the particle just by overexpression. But if you can specifically add the RNA into the budding particle using that Cy packaging signal, you can dramatically increase the amount of guide RNA packaged up to over threefold. So this system that we developed, we term Nanomedic for membrane-derived extracellular vesicles for the delivery of macromolecular cargo. So the advantages of this system is that it's protein-based and transient with no DNA intermediate. And again, the key features of this system are that we can incorporate Cas9 by chemical ligand-induced dimerization, and we can specifically enrich the guide RNA content by adding a packaging signal. So this nanomedic particle on the right figure, you can see that it has the envelope for cell targeting. GAG serves as the beacon for recruitment of both Cas9 protein and the guide RNA. And you have your kind of minimalist protein delivery vehicle for delivery. So how do you make Nanomedic? Nanomedic is produced by transfecting plasmids expressing different components of the Nanomedic system. So you transfect them into HEC293 T cells. In the beginning, we were using adherent HEC293 T cells and a lipid transfection reagent. And after 36 to 48 hours, you would get secretion of these nanomedic particles into the media. And all you do is you harvest the media and concentrate it. And now you have your nanomedic particles that you can inoculate or just simply add to cells. So the image on the top right is electron microscopy image of our nanomedic particles. And we found that the diameter range from about 130 to 160 nanometers in size. So just to go into some applications of Nanomedic in various cell types in vitro and also an in vivo application, we tried delivering Nanomedic onto T cells. So these are actually Jurkat cells. Jurkat cells are not easily transfected by lipid reagents. We wanted to target CCR5, which is exon 1 of CCR5, which is the co-receptor for HIV-1 entry. And what you can see in the left figure is that we added increasing amounts of Nanomedic to these T cells. And as you increase the amount of Nanomedic, you get increased DNA cleavage, indicated by these two cleavage bands on the bottom. And on the right side is just a bar graph depiction of the cleavage activity, or the indels introduced. And you can see there is a dose-dependent increase in mutation frequency as you increase the amount of Nanomedic added to these cells, over 50%. So this is one example of delivery into T cells. We also looked at delivery of Nanomedic into iPS cell-derived cortical neurons. So in this example, we are targeting SAMHD1, which is a gene involved in Aicardi-Goutieres syndrome, an autoimmune disease, and we used 2 guide RNAs to target exon 1. So these cortical neurons were differentiated from iPS cells by neurogenin 2 overexpression, which is a pretty efficient way to induce cortical neuron from iPSCs. So in the right, you can see this is, again, a T7E1 assay, and the bottom bands that appear in the blot image are cleavage products indicating indels have been introduced at the target site. And you can see both guide RNAs had about 36% mutation frequency induced in these cells. So Nanomedic got in well to these cortical neurons as well. One of the more important aspects of our system is the safety, which I was mentioning earlier in the presentation. So we use Nanomedic to target a site VEGF-A in HEC293 T cells. And the guide RNA we chose has been a reported promiscuous guide RNA, which has unknown off-target activity. So the on-target sequence is indicated in green here, and the off-target is in red. So you can see there are only 2 base pairs mismatched, and these are kind of distal base pairs from the PAM sequence. So when you deliver Cas9 and guide RNA in plasmid form, you can see on the bottom left figure, the on-target cleavage of the intended site is about 30%, but the off-target cleavage indicated in red here is pretty high too, over 15%. And however, when you look at Nanomedic delivery, the on-target cleavage is just as good as the plasmid delivery. Sorry, this is in HEC293 T cells, but the off-target cleavage is virtually eliminated. So this data indicates that you're getting specific on-target cleavage, but reducing the off-target non-specific cleavage. And the on-to-off ratio is over 60-fold, indicating that you are getting high specificity when you deliver Cas9 in the protein form. So with that, we're pretty confident that these particles are active and comparable to DNA delivery of the Cas9 and guide RNA. And so we then looked at Nanomedic inducing high editing efficiency in DMD patient iPSCs. So we looked at 2 different DMD patient iPS cell lines, either with an exon 44 deleted or exon 46 to 47 deleted. So with the strategy for DMD patient cells, you can induce the exon skipping of exon 45 by either targeting the splicing acceptor or splicing donor site of exon 45, and that would restore the open reading frame of the dystrophin protein expression. So when you look at this, the top left figure shows just the splicing acceptor site and the mutation frequency at the splicing acceptor site. If you're adding Nanomedic to target the splicing acceptor site, either by itself or in combination with a splicing donor Nanomedic, you can see that you get high indel frequency induced over 60%, and is pretty similar in both DMD patient lines and also in a healthy donor line. Similarly, with the splicing donor site targeted, you can get high efficiency over 50% targeting the splicing donor of exon 45 alone, or even if you combine it with the splicing acceptor site, you're still getting high indel induction. And again, for the patient cells, both patient cells, and also for the healthy donor. So the bottom left figure shows the PCR amplification of the genomic DNA region of interest. So if exon 45 is removed by simultaneous targeting of the splicing acceptor and splicing donor site, you should see a smaller PCR product. So that's indeed what you see when you add the splicing acceptor and splicing donor Nanomedic. So we saw up to 36% of the genomic DNA deletion of exon 45 in 2 patient cells and also in a healthy donor cell. And the bottom right figure shows kind of the bar graph showing a summary of the exon 45 deletion from these 3 cell lines. So it's pretty consistent across 3 different iPS cell lines. So once we obtained these edited cells, we differentiated them to skeletal muscle cells and looked at dystrophin expression. So this is, on the left side, you can see RT-PCR of cDNA extracted from these skeletal muscle cells. The bottom left shows kind of a bar graph showing the exon 45 skipping efficiency. So basically, if you target the splicing acceptor site alone, the exon skipping efficiency is only about 30%. Targeting a splicing donor site had really little effect on the exon skipping. And however, if you targeted both the acceptor and donor sites, you can see that there was a synergistic effect where you have over 90% exon skipping. And if you look on the right and you see the Western blot analysis, you see protein expression of dystrophin restored in these cells with highly efficient exon skipping when using a combination of the splicing acceptor and splicing donor Nanomedic. So I think this data shows that you can highly edit iPS cells, and that when differentiated to the cell type of interest, they show the expected phenotype. So then we looked at in vivo delivery of Nanomedic using a luciferase reporter mouse. So in the top left schematic, you can see that this reporter mouse has luciferase knocked into the ROSA locus. However, the luciferase gene is interrupted actually by exon 45 of human dystrophin. So if you can induce the skipping of this exon by adding the splicing acceptor and splicing donor Nanomedic, similarly to what we did in the iPS cells, you can restore luciferase expression. So if you get luciferase expression, that means that exon skipping has occurred. So if you look at the bottom left, we injected into the gastrocnemius muscle Nanomedic for the splicing acceptor and splicing donor sites, and we did this in 5 mice. And looking at the kinetics of luciferase expression, you can see that luciferase expression happens pretty early on, within a few days, and it kind of plateaus. So it plateaus and it's sustained for 160 days. And these results indicate that you have permanent disruption of the splicing acceptor and splicing donor site of human dystrophin exon 45 in this reporter system. So when we sacrificed the mice after 189 days and looked at the RT-PCR, we could see that exon skipping occurred at about 7% from samples taken from 3 mice. That's indicated in the top right figure. And if we looked at the genomic percentage of genomic deletion in the bottom right by deep sequencing, we also confirmed that there were deletions in up to 6 to 7% of the genomic DNA analyzed. So the deletion pattern was mostly below 50 base pairs in deletion, but we did have some larger DNA deletion of about 130 base pairs, kind of indicating that both that exon 45 was removed from this reporter system. So with that, we were able to show not only in vitro delivery, but we were also able to show in vivo delivery into this reporter mouse. So we tested various cell lines, and we found efficient delivery in all of the cell lines indicated. We also tested multiple endogenous loci and found efficient editing. So once we established that Nanomedic worked, we wanted to develop a culture system that was suspension-based for more scalable production of Nanomedic. And I'll go into the development of how we did that using electroporation. So our previous system used adherent cells, adherent 293 T cells. We also used fetal bovine serum for the media and a lipid transfection reagent. The problem I had when I was trying to scale up the Nanomedic production in this adherent cell line is that the adherent cells, they came off the plates really easily. It was hard to get even seeding density when I went to larger plates. We wanted to remove the fetal bovine serum to make it xeno-free, to make it applicable for more potentially clinical application later on. And the lipid transfection reagent, there wasn't much room for optimization of delivery of your DNA using this. So we found that just technically it was difficult using this adherent system. So switching to a suspension HEC system available from Thermo Fisher that can be grown at high cell density, we also switched to a completely different chemically defined xeno-free media, which could be GMP compatible. And we also wanted to move away from lipid transfection to electroporation using the MaxCyte platform, which is shown in the bottom right machine. So we wanted to hopefully achieve scalable production of Nanomedic, which was easier technically to handle and was also applicable for clinical application. So, I mean, the pictures on the bottom show, okay, if I was generating adherent Nanomedic from adherent cells, I'd have a lot of plates like indicated in the bottom left. The shaker flask, I could minimize the space taken in the tissue culture hood and also reduce the handling of these cells. So that's what we were aiming for in the beginning. And just the electroporation protocol we used was we expanded our HEC cells and passaged them the day before electroporation. On the day of electroporation, we exchanged in MaxCyte buffer, which is just a salt solution and a universal buffer for all cell types. We then mixed the cells with our plasmid DNA for Nanomedic, added them to the cuvette, and electroporated. After electroporation, we rested the cells for 30 to 40 minutes at 37 degrees to allow for membrane recovery, and then we cultured the cells as normal for 36 to 48 hours and harvested the Nanomedic particles. We would then inoculate them on reporter cells and check the flow, the GFP expression by flow cytometry. The initial experiment we did, however, was we checked GFP DNA plasmid expression, and we wanted to compare if electroporation was better than lipid transfection in these suspension cells. So what you can see here is just GFP expression. This is a histogram plot using two different MaxCyte energies. So it's a HEC protocol and optimization 4 protocol. These are two somewhat lower energy protocols available on the electroporation platform. And we compare that with the lipid transfection reagent that came with the LV-Max cells. So what you can see is with the MaxCyte system, you can get nearly 100% of the cells expressing GFP, and the mean fluorescence intensity was shifted far to the right. And I mean, these cells were really green after 48 hours visually by eye. With the lipid transfection reagent, you can also get high efficiency of GFP plasmid delivery. However, the expression intensity was a lot lower than electroporation. And not only that, if you look at the viability of the cells was around 80% after 48 hours with electroporation, versus 40% with the lipid transfection reagent. So we found better delivery efficiency and higher viability using the MaxCyte platform. So next, just to kind of to summarize the process again, we electroporate our Nanomedic plasmids into the suspension hex cells by the MaxCyte instrument. We would harvest our Nanomedic particles and then inoculate them onto reporter cells. And then if we get GFP expression, that indicates that Nanomedic was efficiently delivered into the reporter cells and active. So this slide shows the optimization of the energy used for electroporating the producer cell to generate Nanomedic. And so what happened was, if you go from low energy indicated by 4 to the highest energy indicated by 10 here, increasing the energy increased the amount of functional Nanomedic particles from the producer cells, which is indicated by the GFP expression in the reporter cells. So the higher energy, the more Nanomedic produced from these cells. Energy 9 and 10 had comparable output of Nanomedic, but the cell viability was a bit better in 9. One kind of challenge we encountered when we were optimizing was that production of Nanomedic resulted in a lower cell number and lower cell viability after 36 hours. And so one way we were able to kind of resolve this issue was to add DNA. So we would electroporate the cells, and after electroporating the cell, the DNA is introduced in the cell, but any remaining DNA would be degraded by adding benzenase. So we added benzenase and to cells electroporated at 2 energies, either energy 4 or energy 9. And what you can see is there you get a recovery of cell survival when you add benzenase at both energies. So there are less cells at energy 9, but compared without benzenase, you do increase the cell number at 36 hours post-electroporation. And this is just showing the producer cells remaining at the time of harvest of Nanomedic. You can see that there are only 6 million cells remaining if you don't add benzenase to the electroporated cells, and you have over 3 times more cells when you add benzenase. So what happens to the Nanomedic produced from the cells when you add benzenase? So the cell number and viability increased if you add benzenase, and actually so does the amount of Nanomedic produced. So if you add benzenase at energy 4 or energy 9, you get higher yields of Nanomedic being produced from these producer cells, as indicated by the GFP expression from the reporter system. So we could increase the amount of Nanomedic by increasing producer cell survival. The other thing we found was by decreasing the amount of Vsvg plasmid, which is the surface envelope used on Nanomedic for cell targeting, we could also increase the production of Nanomedic and its delivery into recipient cells. We didn't really look deep into the mechanism, but we believe by reducing the Vsvg amount, you're reducing the amount of empty particles which might be coated with Vsvg protein that might compete with all-in-one kind of Nanomedic for delivery. So we got more efficient production and delivery of Nanomedic when we reduced the amount of Vsvg. So after optimizing conditions for Nanomedic, we then looked to produce Nanomedic by flow electroporation. So here you see kind of a range of MaxCyte's cuvettes from the left to right. From the left is the smallest volume. You can work, as low as 15 microliters, and to the right you see with the CL2 you can go as high as 100 mLs for electroporation. We were actually working for the optimization at the OC400 range, so 400 microliters of cells and DNA, and we were using approximately 60 million cells for electroporation. So after identifying the DNA concentration and the electroporation energy, we could then apply those same conditions for the Cl2. So flow electroporation works by the cells being in a sample bag and flowing 3 mLs at a time into an electroporation chamber and then sent to a collection bag. So just to give you an example of how flow electroporation looks in terms of consistency and scalability, if you have 100 mLs, and this is looking at K562 cells transfected with GFP plasmid DNA, so each 3 mL fraction that was electroporated was collected and then analyzed for GFP expression viability. And you can see 30 separate fractions that were electroporated had very similar GFP expression and cell viability. And when you compare that to the small scale electroporation with 40 million cells, you can see that the expression viability were also similar. So going from 40 million cells to 6 billion cells with the Cl2 gave very consistent and reproducible results. So in terms of our system, how we worked the flow of the experiment was we scaled up production of our suspension hex cells in large Erlenmeyer flasks, and then we performed flow electroporation of 1.2 billion cells. We treated with benzenase because we found that it helped survival of the producer cells, and then we cultured the cells for 36 to 48 hours in the presence of a dimerization ligand, and then we harvested Nanomedic, and we analyzed the amount of Cas9 being incorporated. So in this slide, what you can see is we compared production of Nanomedic using 48 10 centimeter plates compared with suspension culture. So each one yielded 480 mLs of culture. And what we did was we concentrated Nanomedic, and then we lysed the particles and performed an in vitro assay to quantify the amount of active RNP being released from these cells using both systems. And once the RNP is released in vitro, we add a PCR DNA template, which would be cleaved in the presence of active Cas9 RNP. So you should see 2 cleavage bands. So on the right here, you see a standard recombinant Cas9 protein. And if you increase the amount of Cas9 protein, you increase the amount of cleavage bands. So we would just compare the amount of Cas9 contained in these nanomedic particles with the standard curve to quantify the amount of Cas9 being released. And what we found was that the adherent system with 480 mLs released roughly 27 nanograms per ml of active RNP complex. If we compare that with kind of a small scale run with 60 million cells and with 1.9 billion cells, there was a reduction of about 34% of the active Cas9 being released. However, we see consistency between the small scale and large scale runs for nanomedic production. And considering that we changed the cell type and we also changed the media composition completely and the delivery method, we felt that the suspension was easier and more manageable if we wanted to scale up further. And not only that, we were able to obtain xeno-free conditions using a chemically defined media, meaning that there could be potential clinical application for this system. So in summary, we developed a xeno-free nanomedic production system by electroporation that could be scalable from 60 million cells to 1.2 billion cells. We found that it was easier to handle, and we feel there's promise for industrial applications. Benzenase treatment, we found after electroporation, did enhance cell viability, and we were able to improve nanomedic yields. So the energy used for electroporation, if you increase the energy, you increase the amount of nanomedic particles produced. And we did feel at the end that electroporation was an efficient method for producing nanomedic. future directions of this project, we think that the cell targeting can be changed of Nanomedic by changing the viral envelope or maybe adding a single chain antibody. The other thing too is that you're not limited to just Cas9 delivery. You can incorporate any protein of interest by fusing a dimerization domain to it. I didn't show it in this presentation, but we were able to efficiently package luciferase protein as well into Nanomedic, and we could deliver that in vivo as well. One could add a viral antigen and potentially use Nanomedic for vaccine production. There are certain aspects of the downstream optimization which are further needed, such as affinity chromatography purification, which would increase the purity of Nanomedic and maybe allow for more efficient delivery. So there are a lot of acknowledgements. A lot of people contributed to this study. Akitsu Hotta, who was my mentor and who really helped to give ideas for the project. I'd like to thank him, as well as Noriko Sasakawa, Mandy Lung, and Yuya Okazaki, who really helped push the revision of the experiments. I had a lot of help from summer interns who motivated me to work harder, and I really appreciate their support, as well as a collaboration with Takeda and for the T-Styra project. Yukimasa Makita and Hiroyuki Hozumi and Naoto Inukai were responsible for the in vivo studies. Jun Komano gave a lot of ideas for when I first started the Nanomedic production, and other researchers like Takeshi Noda, who helped with the electron microscopy, and Yuko Fujita. So with that, I'd like to thank you, and I'm welcome to take any questions or comments. On the right, I also want to point out that we have a research spotlight on this study that is downloadable from a link that you can find. And there's a link to the manuscript itself. So again, thank you for your time, and I welcome any questions or comments. Thank you.
Great. Thank you, Peter, for that informative presentation. So at this time, we will open it up and use the remaining portion of the session to ask Peter any questions the viewers might have. So if you have a question and you'd like to ask it, please do so now. Just as a reminder, you can use the Ask a Question box located on the far left of your screen. So let's go ahead and get started. So the first question that has come in, Peter, is how does the Nanomedic transient protein delivery system compare with other systems that are currently available?
Well, I mean, if you compare the Nanomedic system with other transient systems, I did try other published reports like of the Nanoblade system where you would fuse Cas9 with GAG, but there are a lot of limitations of that system because you have to cleave Cas9 using a GAG protease to liberate the protein, and that sometimes cuts Cas9 itself. So I think with the other systems, I didn't find that the guide RNA packaging too was efficient. So at least with other published reports, I wasn't able to find both being incorporated into the particles efficiently.
Okay, great. Next question: how does Nanomedic compare with AAV delivery?
I think it's kind of difficult to compare both. Nanomedic's a protein-based delivery system, and AAV just needs a single DNA vector to express a large amount of Cas9. We did some preliminary studies where we found the kinetics, at least, of Nanomedic were faster, but overall, I don't think we fully optimized the conditions for kind of a fair comparison. So, yeah, we need to develop it further.
Okay. Great. Next question is, are the tropisms similar to lentiviral Vsvg, and do you need to use immune suppressor for in vivo use?
The tropism is exactly the same because this particle uses Vsvg for delivery, so it'll target low-density lipoprotein receptor. In terms of immune suppressor for in vivo use, unless the patient has been exposed to vesicular stomatitis virus or maybe HIV beforehand, I don't think there would have to be an immune suppressor.
Okay. What about comparing with just electroporation the ribonucleoprotein particle, and that is Cas9 protein complex in solution with genomic RNA?
Oh, with guide RNA. So, we did do this. It's one figure in our paper, but what we found was, for example, if you electroporate the RNP in solution, you have to add a lot more. So we compared like 10 micrograms of RNP being electroporated versus 200 nanograms being delivered by our Nanomedic, and we had actually higher delivery of RNP into iPS cells with our system. So we found that it's either the same or it's been better in some cases than electroporation.
Okay. Next question. Can Nanomedic be administered in vivo by intravenous injection?
We did do some preliminary studies with that, trying to deliver a luciferase protein intravenously. We couldn't observe luciferase signal intravenously. So for now, at least, it's just through localized injection. But we will optimize in the future probably for more systemic delivery.
Okay. What is the cell target specificity of the Nanomedic system?
So kind of related to an earlier question, so we use Vsvg, which targets low-density lipoprotein receptor, which is expressed on virtually all cell types. So there isn't a specific cell type that we're trying to target right now. In the future, you could change the viral envelope or maybe add an antibody to target a specific cell. So that's something that could be engineered later for more cell specificity.
Okay, great. Next question. What other types of cargo could be loaded into Nanomedic?
So, I mean, that's a really interesting question. We did load Cas9, and ideally you could load other genome editing tools, or perhaps something like prime editor, which is actually quite large to add by DNA plasmid. So in terms of gene editing, you can add a variety of nucleases or other editors. You could probably add fluorescent proteins for labeling cells, at least transiently. And also for vaccine development, you could load them with viral antigen, which could be an option too.
Okay. Next question. Would it be possible to load a DNA donor template into Nanomedic for homologous directed repair with CRISPR Cas9?
I don't think it would be possible to load the donor template inside the Nanomedic particle. A similar system called Nanoblades was able to kind of complex the donor template outside of the particle by adding polybrine, and then they could co-deliver it together. So we did try a similar approach, which wasn't in the study, and we were able to get up to 9% HDR efficiency in iPS cells by pre-complexing Nanomedic with a donor template. So it is possible. It just, I don't think you can package the donor template inside Nanomedic. It'd have to be externally kind of packaged. Yeah.
Okay. Are there other applications in addition to CRISPR Cas9 genome editing?
Other applications to genome editing, right? So kind of similar to what I said before for maybe labeling the cells or vaccine development. I guess for any kind of molecule you want to deliver, maybe you can even deliver the Cray recombinase. So I mean, any protein of interest could be loaded into Nanomedic. I think it's more advantageous to try to load larger protein cargo that you wouldn't normally be able to deliver by DNA plasmids.
Okay. Next is, how long did the electroporation optimization for Nanomedic production take?
Oh, this one, it wasn't too long. I think I was pretty able to optimize at the small scale in the OC400 within 2 months. The only limitation I really had was I didn't have too much incubator space with enough shakers to do more at one time. But I was able to optimize and scale up within, I would say, 2 months up to the CL2. So it was pretty fast for the optimization.
Great. Looks like next question. What are some other technical challenges in producing Nanomedic?
I guess the technical challenges I found were the purity of the particles, because when you concentrate them, it's not just Nanomedic, but you're concentrating exosomes or potentially some other cell debris. So I think purification by affinity chromatography would be pretty important to remove any cellular debris or exosomes from the Nanomedic preparation, and also maybe any residual DNA that might be left over. So, and also, I guess for Nanomedic, another technical issue could be the more you scale up, it was just more of the culture conditions for large cell number, which we didn't have the capacity for in an academic lab at the time. So maybe those kind of technical issues came up during the optimization.
Makes sense. Okay, next, and looks like final question. Is there a risk of other cellular components being accidentally incorporated inside of the nanomedic particle during production, which potentially may affect the target cell?
So we did do a proteomics analysis of the nanomedic particles. We did find some kind of cellular host proteins, but considering that they're proteins being incorporated, I think the delivery of those proteins would be pretty transient and not in a high concentration. So I don't imagine that there would be any kind of toxic issues with that.
Okay, and actually another question did come in. Have you looked at the content of the nanomedic? And if yes, were there other proteins? Sorry, Peter.
Oh, sorry. So I guess similar to the last question, but so in particular, what kind of proteins? I guess a lot of exosome markers were in there. Of course, our nanomedic components like GAG were highly incorporated, but I don't think there was anything overwhelmingly like incorporated more than the overexpressed proteins we had in there.
Okay. Well, that looks like all the questions that we had come in. So thank you very much for, for answering those, Peter. I wanted at this point to remind our audience that the recorded version of today's talk will be made available soon after this live session, and you will be notified via email when it becomes available. Also, any questions that are unable to be answered today, which I think we got to all of them, but those that do come in during the on-demand session will be answered using the email address you provided during the time of registration. So before we go, I wanted to take a little bit of time to thank Peter for his detailed presentation, to thank our customers and industry partners for allowing us to share their important work with a larger community, and to thank LabRoots for their supportive webinar team for organizing and promoting our event. And lastly, I would like to thank you, our audience participants, for joining us today and for their interesting questions. So this concludes our session for today. Until next time, take care.